Thanks for having me again. The conference 2 years ago was already a fantastic conference, and I really enjoyed that. And so I'm extremely excited to be back. Yeah, I mean, it's in many ways a rerun of last year. So last year, or 2 years ago, Jumpai presented before me. So this was already a perfect lead-in to the topics that I would speak about. As Victoria already mentioned, one of our mainstays has been optimizing the ability to localize sounds in space. Yeah, and over the past 2 years, we've made a lot of improvements on this front. But let me first introduce to you a bit the overall topic. Of course, we all know that rodent vocalizations during social interaction are very important, and they're very important for many reasons. There's, for example, mother-pup interactions where pups vigorously vocalize when they're separated from the nest. I'll talk much more about this later today. Then, there's vocalizations being used during courtship and territorial disputes, and so many publications have looked into this and provided detailed analyses of these. But more importantly, maybe for the human side, the mouse models are often also, the vocalizations in mouse models are often biomarkers for psychiatric disorders. So, for example, today I will be speaking also about the SHANK3 knockout mice, which is a model for autism spectrum disorder. And then there's also FOXP2 knockout mice, which are a model for language production deficits. So challenges in studying social vocalizations, as we've already heard in the previous talk, are that they're on the one hand ultrasonic, so usually we can't hear them. They occur in close proximity, so precision is very important. There's often occlusion and echoes. So in that sense, we're really interested maybe more in the sound field, the whole sound field, rather than just a single estimate, like Quattro just indicated as well. So researchers have generally been quite creative about circumventing this problem. So, for example, there have been study of interactions of only same animals. So, for example, only male mice or only female mice or so. But of course, or other ways, for example, devocalizing mice or anesthetizing one of the mouse or separating them physically so that who is actually vocalizing at a given time. But we're more fans of solving the problem bottom up, so really solve it on the let's say physics level. So the earliest study that really looked into this in detail was a study from Neunübel et al. In 2015, who reached an accuracy of about 38 millimeters. So that was the median error. And then a few years later, both Junpei's group and our group advanced this quality to about 14 millimeters. But if we continue on this path of adding more microphones, which was the key in both of those studies, then at some point, the mouse will be very much encircled by, bulky microphones, and they also can be quite costly, right? So all of you using AV soft or other microphones know that each microphone costs maybe 2,000 euros or so. So if we, go further, then this becomes at some point quite prohibitive in cost and size. So the direction that we went into is to use instead acoustic cameras, which can provide an effective solution for this. And in 2023, we published one paper where we showed that we can get down to about 3 to 5 millimeters in localization accuracy, using this approach. So I would now show you what happened in the last few years in terms of acoustic cameras. So here you see the so-called SoundCam 3 Ultra sensor, which is our current, workhorse, so to say. So this one is an acoustic camera that has a huge number of microphones. So this one here has 176 microphones, which you see are arranged in this pattern on it. So each hole here on the left is a microphone. Very importantly, we have an integrated alignment with a visual camera that is sitting here in the center, and which very importantly also, is able to perform infrared imaging, which is particularly in rodent interaction essential. One thing that's very important also that it provides real-time raw data access, which I'll show you later is very important for our concrete application. Also importantly, it has a very compact form factor. So you see here it's just 5 by 20 centimeters. So you can essentially conveniently hang it over most enclosures or the action spaces that you might be interested in. Also interesting, it has a processor integrated that can assist you in computing sound fields, for example, live, so-called FPGA processor. And very importantly, and this has actually also improved a lot in the last years, is that they now come in at very low price points. So now we're talking about roughly 7,000 euros for one of these cameras. If you would kind of scale up the other microphones, then you'd be talking about more like 100,000 euros or so per setup. However, of course, these microphones have an issue. So if you look, at the very high quality microphones like the AVSoft CMPA-16, which has, a very beautiful noise level across high levels of frequency, and you compare this to one of the internal microphones that you have, in one of these sound camps, then you see that particularly at the high frequencies, you have a substantial amount of noise being added. But we can use the microphones, the high number of microphones, to overcome this to a certain degree. And as we will show this, to a degree that you can use it very well for studying rodent social interaction. So this is done by the so-called beamforming algorithm. So it's actually a very simple algorithm. So if you see here on the left, if you have two sources of sounds, and they provide sounds and they arrive at all of your microphones, then one of the key aspects of distinguishing these sources is that they arrive at different delays. And here you see the signals, arriving at each of the microphones with, in kind of their natural time base. Now, if you shift these delays to match source one, you just compute the path to source one, and you can see that now all the signals from source one nicely overlay. And if you summate them, then you see that now you get a substantial increase in the in the signal size. The same principle that Jumpa and colleagues have used in their USV camp. So now we can, first of all, estimate analytically what should be the advantage of, combining many microphones. And so here you see that, if you have 64 microphones, then I've gauged this so that, we hit the 5 millimeters localization accuracy, which is what we actually find. And you see if we go to 176 microphones in the current iteration, then we should be able to go down even lower. So roughly here between 2 and 3 millimeters in precision. But secondly, very importantly, you can also perform denoising. Oops. I guess you guys can also see this Zoom thing, but I can't see my mouse. So I can't move it out of the way. Okay, we'll have to live with that. So you see here that the noise in the microphones is, it's independent. It's essentially just thermal noise in the microphones. And so this thermal noise is, getting reduced by the number of microphones as 1 over square root of n. So that means, after beamforming, if you have 64 microphones, you already have an 8-fold decrease. And if you have 176 microphones, you get a 13-fold decrease in the noise level. And you see this here for, our previous generation device, the CAM64, or the L642 actually. And there you see, this big difference in the quality, or the reduction in the noise at high frequencies already. And, I will show you some vocalizations in this context in a moment. So we've also estimated, for the L642, so a device that has 64 microphones, the accuracy, and we've done this with a speaker to get, some ground truth estimates here. And you see here for different locations in space, you see the location in space is essentially the red dot, and you see the blue dot is, our localization centers, and you see that they very precisely match, the original location of the sound. And if we then, estimate this as across different intensity levels, that you see that, in the range of 70 to 90 dB, we have this range of about 3 to 2 millimeters in median absolute error. At 60 dB, that starts to fall apart. But please notice that, the typical vocalization amplitude of mice is around 80 dB SPL. So typically we don't see a big problem in, vocalizations being too quiet in that sense. So here's an example of what this looks like in practice. So, like in other studies, we have here frequency converted the vocalizations so that you should hear them. And here on the left, whenever you see a yellow dot, then this is, the localization of a sound. But you also see kind of the, the sound field estimate on top of it. And you will see that this is kind of very narrowly centered around the snout of the mouse. Yeah, and so, clearly also like Guato was already just showing, this technique of beamforming and using many microphones can very effectively provide precise localization, in these conditions. So, actually nicely following up on one of the questions that was just asked before, one of our main interests is to turn this into a real-time audiovisual tracking system. And this is where Ruben, who is unfortunately covered here a little bit by the images here. has been making major contributions over the last years. So the system in our hands looks as follows. So we have this acoustic camera just placed above the cage. And one thing I really want to emphasize is that the alignment of the camera and the microphones is really handy, because in this way you never have to worry about aligning them in a precise way, which was actually an issue in our previous setup. Yeah, where if you want to achieve millimeter precision, then of course everything has to be millimeter precise aligned, and that can be a bit annoying also, because your camera can have a relative angle to the microphones and so forth. So we're very happy with this integrated setup. So we can then obtain live video from this device, typically at about 50 frames per second, and live audio from all of those. So we get those at 200 kilohertz. So at this point, you still see the individual microphones, and you see that the SNR is here, not yet very good, but this is still without beamforming. Now we can combine this, and we use video tracking based on sleep, a package very similar to Deep Lab Cut. And then also, I will show this in a more detail in a moment, but we also use this tail re-identification to perform long-term tracking. In addition, you see the sound field estimation. So this is here, you see the red dot on the, on one of the mice, which is at this moment, where a vocalization is coming from. And then we can perform now beamforming to each of the snouts of the mice. So very important in our approach is that we use, the tracked positions of the mice, so the snouts at each point in time, in order to perform beamforming to this location. So, this also addresses a point that was just raised by somebody before. In this way, we can essentially circumvent this problem of echoes that might occur through the walls. Yeah, because the echoes might look like the source of a vocalization on the wall. And if you first look at the sound field and then, you estimate the location, you might actually erroneously place it on the wall. But our approach is inverted here. So we first track the mice, and, then we use those positions as seeds for our beamforming. And now if you look here at the beamform streams, then you already see that the signal-to-noise ratio has markedly increased. And now we can compare the intensity for each time frequency point in those spectrograms in order to assign it to one of the mice. And so if you look at this here, then you now see that we get essentially per vocalization or even subpart of the vocalization an assignment to one of the mice in the interaction. And so one thing, like I was mentioning, so this analysis is running on a GPU in the computer, and that is essentially piping the data live, or the system is piping the data live into the GPU and is performing the analysis. So analysis here meaning visual and acoustic tracking live, and is also saving the data. One thing that I'm going to go into in a moment is that we can use this live aspect to perform real-time feedback. But one thing I first want to mention is that the system then allows allows the user to conveniently look at some of maybe the most obvious analyses that they might be interested in. So for example, on the very left here, you see in H the vocalization rates per animal. You might have perform syllable analysis. So very similar to what previous speakers have mentioned, we've also kind of moved away from the classification approach, but we kind of more buy into the idea that it is still valuable to quantify the syllables in terms of a lot of acoustic properties. So for example, then you might have vocalization frequencies for different mice here. Then you have, you can get a spatial vocalization map. So, that's a very, the direct readout essentially of where our vocalization is coming from. And then one thing that we really like are these so-called relative spatial vocalization histograms. So here on the right, you can see how, where other mouse were located at the moment where a given mouse was vocalizing. So the given mouse here is, indicated by the green, blue, or orange body in the center. And essentially, the histogram is then the relative spatial position of other mice relative to this mouse. So here, I just want to quickly show, how this feedback works. So, our idea is that we can have here for example, target area. We can say, okay, if an animal, if a particular animal is making a particular vocalization in this kind of a target area, then we want to provide feedback. And you see this here in the middle. So you see that these yellow vocalizations are essentially, you only see it here in this one location. So here in this moment, this yellow vocalization is created by us, by a speaker, and that's happened, triggered by this vocalization before. Yeah, and here you see it also. So you see here the trigger is being, these are triggers, and then we provide feedback here at particular moments. And very importantly, this can happen now at relatively fast speed. So you see here that the pipeline speed depends on various things. So sleep is the visual tracking, green is the beam forming, and DAS, the deep audio segmenter that was also mentioned yesterday already, we use for detecting vocalizations. And you see each of those, run at about 10 milliseconds each, and then we get a total kind of round trip time of the system of about 40 to 50 milliseconds. And at that rate, our pipeline is updating at about 20 frames per second, meaning that we can provide feedback after a delay of maybe 100 milliseconds roughly. Similar to what was said before, this kind of approach allows us to also show vocalizations, assign vocalizations that are happening at the same time. So here in the bottom, you see, this green rectangle around one of the vocalizations. You see localizes to one mouse. These are actually pups, so even smaller than adult mice. And on the right side, you see that if we now select a different time frequency range here, then you see that it localizes to the other pup. Yeah, so that's really, of course, a great advantage of the sound field estimate that you can do this simultaneous localization. Then one thing I want to mention is that, because a lot of you are, of course, users of the great Live Mouse Tracker, and the Live Mouse Tracker uses RFIDs, and essentially, because our mission here, so to say, is a little bit to have, a widely deployable single device system, and for this, not using RFIDs is an important kind of constraint for us. And so essentially, we have to perform, first of all, markerless tracking. So, what you see on the right is, the current performance that Ruben can achieve using sleep on multiple animals. And so this is a video that runs for about 10 minutes. I mean, I won't have time to see it all, but that is roughly our current state. That, for 10 minutes, you see that it tracks very reliably. It maintains identity over the animals, and you will see here, for example, even if they come very close or they completely overlap, it doesn't lose identity over this. And essentially, my conviction is that at the pace at which AI is evolving at the moment, I would say that whatever a human can track visually will probably be trackable by these systems in the near future. Some of you maybe know this tool called Lightning Pose. This, for example, extending tracking, through short timelets or tracklets, what they call it. But on the other hand, of course, in principle, we want to be sure that we can track over time. So we also have a system of re-identification. You also see this here. So, some of the mice, as you can see here, have a tail marking. So these markers are, of course, less permanent. They're also less invasive, and in that sense, more flexible, right? You can remark mice or have different markings based on, your daily needs. They work in more environments, I would say. So, for example, we have a home cage, so we don't need to put RFID sensors underneath it. And I would also say that under certain conditions, they also allow faster re-identification and close interaction. So, by what I mean by this is that if two animals are close to each other on an RFID sensor, then you don't get a read from it, or no identity. But but in an image, you can, as long as the tails are visible, you can still re-identify even if they're rather close. And so in principle, this technique is also possible in non-flat habitats. Yeah, so if you have some elevated platforms or so, that would be possible, which I think might not be possible with RFIDs. But on the other hand, of course, we are more prone to occlusion, right? So RFID would work if you have a shelter, whereas we would not be able to work inside a non-transparent shelter. But the way that we do this is that we have these, multi-stripe barcodes on the tail, and Ruben has developed some machine learning tool, also variation autoencoder based, to reassign them. And here you see some, intermittent data from him, where you essentially can see that. So in this case, we only had, the 000 1001 and 1100 label, and you see that you have a very high probability of recognizing these again. And this was actually based on the Sorama L64 camera, which had a relatively low resolution and JPEG compression, which you see here. You see this classical issue of JPEG compression that it gets kind of blurred. And if you look now at our current quality with the new sound cam, then you see already that quality is much improved. And also importantly, we get raw data, so that means this compression issue is not there anymore. Okay, so with this, I just want to conclude the first part of the talk where I try to show you that this integration of sound cam with machine learning, acoustic cameras with machine learning allows you to get towards a live system of audiovisual tracking that allows potentially also real-time feedback. So we've been using the system in the past years, and so essentially I want to show you some case studies where we see value in such a system. So one of the applications that I've already shown last time is this competitive interaction between mice, and I just want to use it as an opportunity to introduce these spatial interaction histograms again. So here, first of all, we might have two mice that are interacting with each other. And so again, if you have here, for example, the male emitter in the center, the blue mouse, then you can see where is the relative position of the snout of the female mouse, while the male mouse is vocalizing. And you can compare this, for example, to when does the female mouse vocalize, and you see this stark difference that essentially the female mice mainly vocalize in snout-snout contact, whereas the male mice vocalize in snout-snout contact, but also, and dominantly, if you add up the numbers, in anogenital contact. Then now we use this system to study 3 mice together, and one of the insights that we had from this was that there's essentially quite a difference in whether a mouse actually partakes in this interaction in a vocal sense or not. So what you see here in the plot between dominant and subordinate is that there in each interaction there tends to be a mouse that is very strongly active, whereas there is another mouse that then another male mouse, sorry, that chooses to not be very active. So we saw this really divide between the mice taking part in this interaction. For female mice, we don't see a lot of vocalizations typically. Yeah, so even the cases where you have here a high fraction are often interactions where there just is not a lot of vocalizations in the overall recording. Yeah, but you see that female mice tend to be rather quiet in these courtship interactions. Yeah, so summarizing this, that we have one male that is pretty dominant in the vocalization behavior in triadic interaction, and female mice are relatively quiet during these courtships. Secondly, we've looked at let's say a relatively popular model mouse for autism research, namely the so-called Shank3 knockout mice. And this is in collaboration with Judith Homberg from the Radboud UMC here, who's an expert in neurodevelopmental diseases, and who granted us access to these mice. So first of all, a quick recap on what the Shank3 knockout means. So the Shank3 gene in humans and mice is a scaffolding protein in the postsynapse. And so you see it here. It's linking essentially different aspects related, different parts of the postsynaptic machinery to each other. And when it's missing, then you have kind of a disorganized postsynaptic site, which then leads to overall reduced dendritic spine density and also reduced excitatory transmission, as you see here, it's associated with NMDA and AMPA channels. If it's missing or mutated, then it is the genetic cause for the so-called Phelan-McDermott syndrome, which manifests itself in delayed development and also intellectual disabilities in humans. And also, it's considered to be one of the few monogenic causes for autism spectrum disorder. And so in mice, there are, and that's why mice or Shank3 knockout mice are actually a very good model, right? Because you have a close genetic basis here that you can exploit. And so so there have been multiple studies, some by Elodie Drapeau and also Fabrice de Chaumont, which actually was part of the original publication of the Live Mouse Tracker. And so what you find is that these mice generally show, stereotypies in their behavior, excessive grooming, and strong escape behavior. However, in the study by Drapeau et al., they emphasize that if you look at dyadic, so two animal interactions, male-female interactions, there was really not a strong impact that they found, and that's the same thing that Fabrice also showed, in his study on this. In particular, they found no significant change in vocalization behavior. But one thing that the live mouse tracker, of course, enabled is to study groups of mice. And so in more interactions, so in particular triadic interactions, shank knockout mice showed some aberrant behaviors. But in this study, no vocalization behavior was really analyzed. So this is one thing that we wanted to address here. And so we all hypothesized that the vocalization phenotype might only be effective if complex social interaction is involved. So here we studied, sort of triadic male-male female interactions, where one or two of the mice, or none of the mice actually, were knockout mice. So we essentially had, wild type knockout, or wild type or knockout. And so the setup is very basic, right? So we have essentially the sound cam above, and then we have the three mice interacting for 10 minutes with a female. And so here you see, our basic results. So first of all, if you focus on the two dyadic conditions, so if we have dyadic wild type and dyadic knockout, that is essentially replicating what these other studies found, namely that there is no significant difference in the amount of vocalizations that wild type or knockout animals make, male mice make, in these dyadic interactions. On the other hand, when you look at the triadic wild type versus knockout, then you start to see a huge difference, right? You see that in triadic conditions, wild type males are much more active, and vocalize much more in comparison to the knockout animals. And maybe bear in mind, or compare this to the study that I previously mentioned about the male-female interactions with these were all wild type mice, where you also saw that one male animal is kind of the dominant vocalizer, and here you see that role then falls primarily to the wild type mice, where the knockout mice in this triadic interaction become quite quiet. And we can also now split this up here by the different subconditions that you have. And here you see that, whereas if you look at individual mice, so every dot is an individual mouse, if you see that, if you have wild type with female, then their vocalization rates go up if they are then paired with the knockout instead. And on the other hand, if you look at the knockout mice, then you see that their vocalization rates go down if they're paired with the wild type. And so, this is also visible then in, the spatial relation between the vocalization patterns of the mice. And you see here that, if wild types are close to a female, they vocalize already more than when they are far to the female. But in both cases, they vocalize more than the knockouts, which actually do not show a significant difference depending on whether they are close or far from the female. And then secondly, we also looked here at the female vocalizations, and you see that also female vocalizations respond to, wild types and knockout animals differently. So again, this is here, the female animals, and you see here these are the vocalizations that they emitted and where the wild type was localized. And you see here that we see a similar pattern that we had before, namely that the wild type animals were in relatively close, so snout-snout type of interaction distance with them. If you look on contrast in the knockout mice, then the knockout mice are relatively randomly distributed. So we don't see a kind of a focus of their location relative to the female when they vocalize. Okay, so summarizing, what we saw here. So essentially, we could confirm that we don't see a difference in the dyadic social interaction, also in terms of the vocalization behavior. But we do see substantial differences in triadic social vocalizations, right? Which we saw both in terms of rate differences, but also relative location of the animals to each other when the vocalizations occur. And one additional result that I have shown here is that we also confirmed there's no changes to the syllable repertoire or the distribution between knockout and wild type, for which we also used the variational autoencoder plus UMAP, which is work by Ruben Versteeg, shown down here. Okay, and finishing up, or maybe I think I'm already out of time. Okay, so because I'm out of time, I think I will skip the last part on these pup isolation calls. But so essentially, we there find that if two pups are close to each other, sorry, if two pups are both separated from the mother, then we see that they seem to significantly modulate their vocalizations in a way that could be interpreted as trying to be more recognizable. Okay, so in conclusion, I hope I could convince you that these acoustic cameras, in this live system, live scope that we're developing, are an interesting research tool that many of you might want to embrace because they provide very high spatial resolution, cover a pretty wide range of frequencies. They also allow this denoising through beamforming, which overcomes the limitations that you usually have in these setups. They're cost efficient and easy to set up. And so I think they open a new level of accuracy for studying social interaction. And then the Schenk and knockout mice, we saw that there's a clear behavioral phenotype in triadic, but not dyadic interaction. And lastly, that the pups modulate their vocalization frequency in competitive isolation, but not in single isolation. So of course, the work was done here by a big team. Most importantly, Ruben Tönnies, who has been working on this for a long time, and essentially vast amount of this work is, of course, due to him. Thanks a lot to Ruben for this great work also in preparing this talk. And then Ruben Versteeg, who's been working a lot on the audio analysis. And I also want to really thank Judith Homberg, who has been a great collaborator over the past years, on this whole development process, but in particular then also on the disease models. And also Lucas Nolas, who you heard speak yesterday, whose interest in this audio localization with acoustic cameras is really what kicked off our interest in it as well. And we really hope that Nolas will eventually commercialize this, because in particular as the costs are coming down, I think this is becoming commercially viable for a lot of labs. Yeah, with this, I would like to thank you